Thanks, Josh. Thanks for this very nice introduction. I'm very pleased to come here for this seminar. This is actually my first time being in Michigan, but so far it's great. So today I'm gonna mostly tell you two stories that moves toward developing differential and also predictive models of single cells over time and space. So the first story is about Dynamo for predictive modeling of single cell genomics. And then I will tell you a little bit on spatial for advanced spatiotemporal model. So for many years, I always interested in moving toward quantitative models of biology. So I want to start with a very theoretical view to motivate how I think about the biology and how my lab will be thinking about the biology. 400 years ago, in order to understand how the planet moves, Tycho made the first comprehensive observation of the planet movement and generated the first big data for astronomer. And then his student Kepler derived the three empirical laws from this big data, which eventually led to the derivation of the unifying theory of gravitation by Newton. So the key question now is that with the big data generated with a single cell and other technologies, can we also obtain predictive models and theories for the biology? Specifically, as a biologist, similar to astronomer who is interested in understanding how the planet moves in the scale over time, as a biologist, we are also interested in how to understand how does a single fertilized cell give rise to entire organism with trainings of cells in a precisely and also spatiotemporally controlled manner. This really dates back when I was a master student, and I was fascinated by this epigenetic landscape proposed by Conrad Washington, a very elegant metaphor that describes how the progenitor cell can give rise to all kinds of terminal cell types. So at that time, I made really extensive efforts, theoretical efforts, trying to quantify the epigenetic landscape. So I started with writing down the differential equation of a set of self-activating and mutually inhibiting network motif. And then I can mathematically map out the epigenetic landscape and even quantify the optimal developmental dedifferentiation and transdifferentiation paths. At that time, I'll just work as theoretical. So I always wanted to actually integrate my mathematical models with the genomics data that just started to emerge 10 years ago. And this is exactly what I did in my PhD and postdoc. So in my PhD, as Josh mentioned, developed Monocle 2, Monocle 3 to accurately infer the developmental trajectory, and also develop a script for learning the causal regulatory network. For my postdoc, and developed Dynamo that I'm going to talk about extensively today. On the other hand, my computational expertise also helped in reporting many breakthrough technologies, including the scRNA-seq, scRNA-seq, the metabolic labeling enabled single-cell RNA-seq, and recently the stereo-seq. So going back to this problem of embryogenesis, now thanks to advances in single-cell genomics, we are able to really capture the precise transition from all kinds of terminal cell, all kinds of progenitor cells to many terminal cell types in a unprecedented resolution. However, the existing single-cell analysis method are rather descriptive instead of predictive. So for example, most of the single-cell analysis approach will stop right at identifying the cell clusters and also annotating the cell clusters with marker genes. So although this big data position us to be able to generate predictive models, the existing approach are not there yet. In the past few years, there are a few attempts that try to achieve this goal. For example, we have pseudo-10 and iron velocity. So for pseudo-10, we can pseudo-temporally alter the cells along a graph manifold to reveal the gene expression along this graph manifold. And with iron velocity, people also figured out the whole kind of model the RNA kinetics to define the time derivative of spliced RNA so that we can predict the local phase of each individual cell. So all these tools, of course, are very popular and give us a lot of biological insights, but they're still descriptive instead of predictive. So for example, for sugarcane, it only tells you the central trend of the cell phase transition, while for iron velocity, because it has to make a lot of undesirable assumption. So the velocity flow often is problematic. Almost 4 years, actually 5 years ago, when I started my postdoc in Jonathan's lab, I wanted how I can go beyond just discrete and descriptive analysis to more predictive modeling. So what I figured out is that on the one hand, we will need more advanced machine learning approaches. And for this, I figured out we will need to go beyond the discrete as RNA velocity vectors for many cells to a continuous and also differential equation. And it's clear to me that once we have such an equation, then we can use it to make money. It will open doors to many normal analysis that are not possible previously. For example, we can use this vector field to predict how the cells evolves over time. We can also use it to resolve the underlying gene regulatory network. We can even make many non-trivial predictions, like we can predict the optimal reprogramming path and the optimal set of transformation factors that enable the reprogramming, and even make in silico perturbation predictions. On the other hand, I also realized that to power up just predictive models, we will need better technologies, better measurements. So for this, I worked with Xiaojie and Chen to implement the metabolic labeling enabled single-cell. This is a new technology that explicitly tag the next RNA with the RNA nucleotide analog for SU. So then you can explicitly identify what's new and what old RNA from the data. We apply this technology to the human hematopoiesis, and when we tag the splicing information from this experiment and performed RNA velocity with SU value. Interestingly, we find that the velocity flow is problematic and quite noisy. You can see the cells, the terminal cell like megakaryocytes, erythroid, and basophil actually moving backward from the spliced based RNA velocity analysis. On the other hand, if we use the labeling data coupled with dynamo, we are able to automatically learn a smooth and hierarchical transition from the HSC firstly into the CMP and then MEP like cells, which is then ramifies into a neutrophil, monocyte, basophil, erythroid, and megakaryocytes. So this nicely recapitulated the known biology of human hematopoiesis, and they also indicated the this kind of data will provide the necessary information to for building these predictive models. We also demonstrate the generality of this approach on a different system. We studied the neuronal activation after the KCL treatment. With the metabolic labeling data, we can reveal a very nice transition of the cells from 10.0 to the 10.15 minutes, 30 and 60 and 120 minutes. Well, if we use the labeling-based information, you can see the RNA velocity flow is quite noisy and most of the time doesn't make sense. So in order to base the model, the analyze this kind of data and build the predictive models. Yes, please.
You know, I meant to ask this, just wanted to know, say a little bit more about the
right. The noise from the spliced RNA velocity comes from a few places. On the one hand, the spliced-based RNA velocity relies on intron rates, but the intron are actually captured by accident. So the majority of the technology doesn't actually try to remove just intron rates, but which we because of mispreming during the in the intron region, we got this introns. So that's why, in general, the splice RNA velocity can be really noisy, especially for genes that are lowly expressed. But there are also other technical issues, like we need to have assumed the transcription, splicing, degradation rate to be constant. Yeah. Thank you. Yeah. Okay. So going back to this, right? We in order to analyze this kind of data and build the predictive model, we then come up. We build this framework called the Dynamo. Dynamo overall has a full major innovations. So, firstly, Dynamo is a general way to model both the conventional single-cell RNA-seq data and also metabolic labeling enabled single-cell RNA-seq data. And for the labeling data, it can be used to estimate the absolute kinetics parameters. By absolute kinetics parameters, I mean the transcription, splicing, and degradation rate has actual physical units. Like we know how much molecule is produced or degraded or spliced over a unit of time. This is because with metabolic labeling data, you actually control the time you label the cell. So that's where you get the absolute kinetics parameters. Then with the kinetics parameters, you can obtain RNA velocity. And then we can take the discrete and also lowest RNA velocity vectors from each individual cell to impute a continuous vector field. And once we have an analytical form of the vector field, then we can do higher order differentials. Like we can do not only RNA velocity, but also acceleration, curvature, divergence, Jacobian cohesion. And as I will tell you later, all this turn, we actually have deep biological meanings. Lastly, we can use this analytical equation to make non-trivial predictions, like optimal paths and the in silico perturbation predictions. The Dynamo framework models the spliced RNA and labeling RNA with 3 different models. For the first model, it's explicitly used for modeling the conventional single-cell RNA-seq data. So we are model the transcription, splicing, and degradation. For model 2 and model 3, they will be used for modeling the labeling data. But the difference is that in model 2, we don't consider the RNA splicing. While in model 3, we actually consider both the labeling and also splicing. At the core of Dynamo, it's ability to tag the paired gene expression and also the corresponding velocity, basically the X and the dx over dt for many cells. And then we can take just discrete measurements for a lot of cells to learn a continuous function f. So you can input the gene expression, output the velocity for any cell states. And this function actually corresponds to the underlying gene regulatory network. And once we have this kind of mathematical description of the cell state transition, you can then give some initial state of this system and then predict how each individual cell will evolve over time in the gene expression space. And in more detail, this vector field is represented as a weighted summation of a series of kernel. So we can reduce this kernel into like a Gaussian kernel, and each kernel will associate with a landmark, like x tilde. And you can see if your state is away from this landmark, the velocity will gradually decay. And in general, we actually only need about 50 kernels to be able to represent a very complicated vector field. So here, n is equal to about 50. So the key, once we have this vector field, the next question is how can we make use of it and to gain biological insights? So what I'm going to tell you next are some mathematical terms you all learned about in your multivariable calculus, but probably never thought about how we can actually connect with the biological processes. The first term is RNA Jacobian, which is defined as the partial derivatives of the vector field respect the state variable. And for Jacobian, if you see you increase the expression of one gene, and the velocity of another gene is increased, then this indicates activation, and vice versa. For the divergence, it corresponds to the trace, the summation of the diagonal of the Jacobian matrix. So it can be used to reveal the sink and the source of the cell state. For acceleration, it tells you the rate of the velocity, so it can be used to reveal the early driver. And for curvature, which can be used to define the cell fate switch, which is very commonly appeared during the development embryogenesis. Lastly, for core, it may relate to the cell cycle process. So in the next few slides, I'm gonna take some simulation and also real data to demonstrate how we can use the differential geometry to gain biological insight. So here I use this toggle switch model between X1 and X2. So these two genes mutually inhibit each other and self-activate. And because this is a synthetic system, so we can actually map out the entire vector field for the X1 and X2. And we can see on this vector field, we will have three fixed points like A1, A2, and A3. The A1 has a balanced gene expression between X1 and X2. This corresponding to a progenitor cell. While A1, A2, and A3 have respectively high expression for X2 and high expression for X3. And you can also find that for each of the fixed points, there is like a electrical basin corresponding to it, like B1, B2, B3. And all the ion velocity flow will actually converge into this terminal, these fixed points. The boundary between the electrical basin is the separatrix. So here, let's look specifically this dashed line where X1 gradually increase and the X2 is fixed to be around 0.5. We can then plot the velocity of X1 or F1 as a function of X1 and get this kind of curve. We can then calculate the partial derivatives of fx1 with respect to x1, and then just get the Jacobian from x1 to itself. And you can immediately appreciate that there is a strong positive peak. This indicates a self-activation. It's shown on the motif in the left. Because we do know the mathematical equation of the system, so we can actually map out the Jacobian for any point in this space and use a heat map to visualize it. So again, you can see the activation from x1 to itself. We can also do the similar analysis to calculate the Jacobian from x1 to x2. You can see here there is like a very deep value that indicates a repression from x1 to x2. And again, if you plot this in the heat map, you can also see the repression from x1 to x2 in the entire state space.
What is the time scale?
Velocity for velocity is instant, and this is a simulation system here. So I think everything is instant.
I get these dates,
Yeah. Yeah. So I mean, we are talking about the simulation here, so everything it's I think is arbitrary, right? But in biological system like here, we are talking about on the 10 scale for a few days, like maybe 1 week. Yeah. So that's why when people are talking about using ion velocity to study tumor progression, that's generally not a good use case because tumor progression involves months, right? It's much longer 10 scale than ion velocity can handle. So for divergence, if it's if the divergence if it's negative, we indicate the local flow is like converging. So this indicates a sink, or biologically this is a terminal cell type, right? Because the cell will differentiate into just a terminal cell state. And if it's positive indicates the local flow is divergent. It means this cell is going out. So it means we will become other cell type. So this corresponds to a progenitor cell. Indeed, if we perform the Whitehead field analysis and also calculate the divergence for the pigment, for the zebrafish pigmentation, you can see, interestingly, in the middle, showing on the bottom, you can see the divergence is positive, while the surrounding cell have a negative divergence. So what this means is that if you look carefully, you can see this the cells in the middle indeed corresponding to like a Schwann cell precursor and proliferating progenitors, and the surrounding cell corresponding to terminal cell types like Schwann cell, Rodolph cells. So this indicates that our method is able to automatically predict the progenitor state and terminal cell states. Another very interesting feature is the acceleration. So because acceleration can turn on much ahead of the gene turn on, so this can be used to reveal the early driver. Here I'm gonna show you how we can to visualize the acceleration velocity and also gene expression for these two master regulators, SPA1 and GALA1, and just like key master regulators for the hematopoiesis. So you can see, interestingly, along the path from HSC to monocyte, the acceleration for the SPA1 peaks at 21 hours, but the velocity peaks at 47 hours, and the expression won't peak until 70 hours. So again, that means that if you see a gene that shows high acceleration at the early time point, this can be used to predict the high gene expression in a much later time point. The real transformative innovation of Dynamo lies in the fact that we are able to gain mechanistic and predictive insight. So for the sake of time, I just here quickly show you that using Iron Jacobian, we are able to identify minimal network between FLI1 and the KLF1 that can be used to explain the early appearance of megakaryocytes. We can also use the most probable path method to predict the optimal path as well as the optimal set of transcriptional factors can convert, in this case, megakaryocytes back to the HSC, and even predict the genetic perturbation effects. Like here, we lock out the PU1, and you can see the neutrophil and the monocyte cells actually move away its current state and move towards to become MEP-like state. So in the next few slides, I'm gonna go through the dissection path and also in silico perturbation predictions. So ever since the discovery of cellular reprogramming, it has become a very fundamental question in predicting what will be the optimal path and the optimal set of transcriptional factors that give rise to cell-fate transitions. Previously, on this simulation system, I used a concept in fishes, the dissection path, to predict the optimal developmental deviation and the transdeviation path. So in this formula shown on the right, which defines the action, basically the VT corresponding to the velocity along the predicted path, and the Fx that corresponding to the Whitehead field. So since now we do learn the Whitehead field directly from the data, I wonder whether we can actually use the same formulation on our data-driven Whitehead field to do the simulation. So what shown on the bottom left is the predicted dissection path from HSC to each of the terminal cell type. And one interesting feature you can identify is that this path is actually not the shortest path, and you can see the path actually follows the curvature defined by the Whitehead field. This is because if we check the action on the top right, if the velocity VT exactly equal to Fxt, that's basically the Whitehead field, then the action will be 0, and this will be the least action path. The D I didn't mention, but the D generally is the diffusion constant, and in this case we set D to be 1. And although this linear path is visualized in 2D, we can actually go back to the original gene expression space, and we can plot as a heat map to reveal the transcriptomic kinetics along the linear path or lab. So what I'm showing here is the lab from the HSC to base of your lineage, and you can see some genes turn out earlier, some other genes are more transient, and where other genes are peak at the very end. So we can then calculate basically an integral that describes the accumulative change of the gene expression along the lab, and then we can define a priority score. And using this priority score, we can identify GATA2, run like swan, just two known transparent factors that previously shown to be related to base of lineage. And those genes are ranked pretty high in my method. So this indicates that using this kind of recension path and also a prioritization approach, we are able to identify key transparent factors that may relate to the cell-fate transition. So next, I'm gonna use this P1 GATA1 network motif to demonstrate how we can use it to perform in silico perturbation prediction. So as we know, these two genes, they have like a self-activation and mutual inhibition, so it behaves like a seesaw model, right? And if you suppress the expression of GATA1, then the balance will broken and SP1 will win. And then the balance will probably move towards the GMP lineage. We can then use the formula shown in the middle, basically multiply by Jacobian with the data X. So here, Jacobian corresponding to, as I mentioned before, corresponding to the gene regulatory network, and where data X corresponding to the perturbation vector. So if you like lock out one gene, then the corresponding gene will be set to be negative value. And if you activate one gene, the corresponding gene will be set to be positive. And the Jacobian is defined for each cell. So we can do this kind of calculation for all the cells in the gene expression state. And we can then basically connect each perturbation vectors data F across the cells to predict how the cell change their cell fate after genetic perturbation. So we then apply this method to perform the in silico perturbation of GATA1 on the hematopoiesis system. You can see initially the cells at the erosory or MAEP lineage. After the perturbation of GATA1, will leave its current state and move towards to become like a neutrophil and monocyte cell state. And this, of course, is consistent with previous experiment reports. We also did this kind of in silico perturbation for many genes, and all of them actually matches with the experiments very well. It is also worth mentioning that this kind of in silico perturbation is very straightforward. You can basically perturb at any time point for any kind of genes, and you can even do combinatorial genome-wide perturbation predictions. So this will provide a very cost-effective way to use genome-wide perturb-seq efforts that was done in the Weissman lab, my current postdoc lab. So I'm gonna, I hope my previous slides show you that my original dream that tries to connect my mathematical model with genomics data is really realized after 10 years in this dynamo framework. And I also show you that we can use the metabolic labeling to power up the predictive modeling, and we use this kernel method to learn the vector field. And importantly, we can also not only predict the cell fate and reveal the regulatory network with this vector field, but we can even make non-trivial predictions like the least action path and in silico perturbation prediction. So the dynamo framework I just told you enable us to perform temporal modeling of the cell fate transition using the metabolic labeling data. However, as I mentioned at the very beginning, the embryogenesis is not only temporally related, but also spatially controlled. Similar to Dynamo, we in order to perform the based modeling on the spatial axis, we will also need to identify the basic technologies that can power up a predictive modeling for the spatial axis. So for this, I figured out we can use the stereo-seq for spatial transcriptomics modeling. Comparing to existing technology like techniques of vision, which doesn't have a single cell resolution because the beads they're using are very huge, right? And also the gaps between the beads are big as well. So this prohibits us to obtain single cell resolution. Well, stereo-seq actually use the sequencing chip, so they can, in principle, capture up to 400 DNA nanoballs within individual cells. So this gives us a subcellular spatial resolution. More importantly, stereo-seq has a large field of view, so we can easily capture entire mouse embryo with a single chip, and it can even extend to 13 centimeter by centimeter field of view. So now with this advanced spatial transcriptomics technology, we wonder how we can achieve single cell resolution and 3D spatial transcriptomics. So for this, I also developed a new tool called the Spatio. And the Spatio has a lot of functionalities. Like we can use it to perform single cell segmentation. We can use it to identify spatial domain and also perform digitalization. Basically, we can automatically define like the spatial layers and columns on a spatial domain. Like we can automatically define the layer 1 to layer 6 in the neurons in the brain. We can also use we can also, of course, perform ligand-receptor interaction, which is like a hot topic just for studying the spatial transcriptomics. And more importantly, we are able to reconstruct the 3D and perform morphometric analysis, basically to predict how the cell migrates in the 3D space. So for the sake of the time, I'm gonna mostly focus on the single cell segmentation and also 3D reconstruction and morphometric vector field part. As I mentioned, using this stereo-seq, we can in principle capture up to 400 DNA nanoballs within each individual cell. And in the original report, we showed that if you overlay the RNA, the nucleic acid staining, I mean the segmentation from the nucleic acid staining with the detected DNA nanoballs, basically just the detected RNA counts. You can see, very interestingly, the RNA signal is enriched in just segmentations from the nucleic acid. This then motivates us to develop an algorithm that can directly use the RNA signal to perform single cell segmentation. And this is actually very beneficial because sometimes doing the nucleic acid staining or other staining will complement your sequencing quality. And this is also like a time-consuming step. So it would be great if we can directly use the RNA signal to perform the segmentation. So for this, we then develop a new tool called the Staro that performs segmentation purely based on RNA signal. So the Staro started with the raw metrics of gene expression on the physical space. We first perform domain identification. Basically, we can separate the domain with high RNA density, high cell density and low cell density, and we then on each domain we can then use 3 complementary methods to perform pre-processing for the single cell segmentation. All these methods will start with like generating a scatter plot where each point corresponding to the DNA nanoball and the color corresponding to the aggregate RNA counts that was detected by that DNA nanoball. And the first method we developed is the EML algorithm-based method, and the other 2 alternative methods, including like firstly doing a Gaussian blur to smooth the data, which followed by OTSU to perform to select the from the signal, or we can use a local Moran's based approach to identify the hot spot in the space to reveal the enriched RNA signal. So the first method is based on a principle approach that models the from the signal and also background signal using a mixture negative polynomial distribution model. We can then use an EM algorithm to basically separate the from the signal from the background signal, and we can then remove the background signal and overturn the figures in the middle, and then you can see in some regions we will have the valleys, and those valleys basically are the points that has a low or zero RNA expression, but surrounded by the beads that has a high RNA content. So we then needed to fill in just points. Basically, we take in we build like a special graph, and then using brief propagation-based approach to filling in just wallets, just missing points. Once we have just process the data, we can then use three different alternative approach, like either the density peak-based method or data peak-based method, or we can, if we do have the staining image, we can use the seed coming from the RNA staining or cell membrane staining to perform the segmentation. And in the end, you will return a single cell data where the spatial location of each cell, as well as the morphology feature of that cell, will also be recorded. So here I'm gonna show you how we can use this method to perform single cell segmentation on an entire most umbral slice, and especially focusing on the heart region. So we first plot the gene expression of MYL7, which is a marker gene unique to the heart. You can see a very specific expression in this heart region. And we can zoom in and for this region, then we can perform the sterile single cell segmentation. You can see indeed this method is able to identify each they're able to identify individual cells, and each of them show different morphologies. I think the real cool thing about the spatial is really this ability to perform 3D reconstruction, and then also novel downstream volumetric and also morphometric analysis. So imagine we want to be able to reconstruct a 3D model from serial sections, and we do have this kind of data now. Sensman collaborate, and basically in this case we use the stereo-seq to profile a continuously sliced Drosophila envelope. So we slice the Drosophila envelope for about 30 slices, and then we can measure with the stereo-seq. So when you do the slicing, actually you lost the relative coordinates of the cell between different slices. We are then needed to develop a method that name the cells back to the original XYZ coordinate. So this will resulting in 3D point cloud, where each point corresponds to a cell, and the color corresponding to the gene expression. But then we can also do more interesting downstream analysis. Once we have this 3D point cloud, we can firstly create like a 3D mesh, basically cover the point cloud. And then we can also divide this 3D mesh into different work cells, where each work cell has at least, at the most, one single cell. We can, because when you do the slicing, you actually have the missing data between different slices. So we then use a deep learning method that map the spatial coordinates to the gene expression. And once you have this kind of continuous model in the 3D, you can then perform continuous slicing. Previously, the slicing was done from the top to the bottom, but now we can actually slice the envelope from the left to the right. So you can observe the spatial gradient across different angles from this continuous model. Another thing we can do is that we can also reconstruct the tissue level merge for each individual organs. And we can start to ask how each individual tissue will talk to each other to do cell-cell communication on the level of tissue and even organs. We can, of course, also calculate many morphometric features, like we can calculate the names, surface area, volume, and cell density, and also the cell distribution in the 3D space. Those kind of analysis are not feasible with just 2D regular single spatial transformatomics. In order to facilitate just kind of 3D reconstruction and also data exploration, we also developed a online web application that allows the user to upload their spatial transformatomics data, and you can use in the browser to actually interactively explore the data. So now with the 3D data that was reconstructed, we can start to ask some interesting questions that are not possible previously. For example, we can try to understand how the organ will expand over time. The organ expansion probably is because of the cell growth, and we can also try to figure out how the organ will shrink over time. Just probably because of the cell death, and we of course model the cell migrations, which is very common during the organogenesis. And lastly, we can even figure out how different small pieces of premature, immature organ converge into a mature one. And indeed, if when we use the spatial, we analyze the data from the Drosophila, we are able to identify all those patterns. For example, the central nervous system actually migrates, and the mid-guard started with 2 pieces will actually converge into a mature one. The next nature question is, can we actually predict how each individual cells migrates during this kind of organogenesis process? And importantly, can we actually identify the gene that enable such kind of migration? Of course, you can do level image. You can try to use level image to track how the cells migrate in the 3D space. But this kind of method cannot give you the transcriptome in real time. So it will be really cool if we are able to actually not only track the gene, the migration of cell, but at the same time the gene expression changes. So here, I'm gonna use this mid-guard as an example to demonstrate how we can achieve this goal. So you can see initially just red cells, just mid-guard cells at the embryonic hour 7 to 9, and you can see they initially have 2 pieces, as I mentioned previously, and it is embryonic hour 10, the cells become a uniform connected structure. So we can take the gene expression change, gene expression similarity, and also the spatial proximity to connect the cells across 2 time points in the physical space. And in analogy, you will realize that the XYZ coordinates from the red cells actually correspond to the gene expression in the ion velocity framework, where this alignment actually corresponding to the velocity vectors. So we can then tag just basically the coordinates of the red cells and the corresponding alignment names to perform vector field learning, and then we can predict we can learn a morphological vector field. So here, the vector field is actually in the physical XYZ space, not in the gene expression space. So the arrows shown here are the actual predicted migration path of each individual cell. And you can even animate this process. So you can see where the bottom cells move up, top cells move down. And if we look very carefully, you will realize that on just the boundary region, the cells actually have very strong twistiness. And this tristan can be used to can be quantified by the 1,000. So we can calculate the 1,000 for each individual cell, and then we can try to connect. We can try to identify genes that show high correlation to the 1,000 across the cells. And in this case, we show the OTP, which is a known regulator of the morphogenesis, indeed have a very high correlation to the 1,000. And we are actually doing CRISPR experiments to lock out the actually not just gene, a few other genes, and indeed we see if we lock out the genes, the organogenesis, the migration of the cell is stopped. So I have told you that using dynamo, we are able to reveal the ion velocity vector field. We are using this spatial, we are able to learn the morphometric vector field. So one is actually in the gene expression space, while the other is in the physical space. So at this moment, these two are actually separate. They are not unified in a single framework. So it will be great if we are able to actually predict both the gene expression change and also the cell migration at the same time. Here, I'm going to use a simulation to demonstrate this concept, and I tell you how we can act, how we are approaching this problem. Hopefully, once we establish success in the simulation system, we can apply this strategy to the spatial-temporally resolved data. So the simulation system showing here is a digital formation. So we know during the mammalian digits formation, right, the embryo started with no digit, but gradually over time and over space, one digit will become to 5 digits. So if we model this process using a 1D system and plot the gene expression of 3 key transcription factors that relate to the digits. So here is SOX name, BMP, and Wnt. So basically, we can create this kind of animation, right? Initially, the x-axis is gene expression, y-axis is gene, x-axis is space, y-axis is the gene expression, and the color is the 3 genes. So you can see initially, the expression is pretty random, but over time, the full 5 digits is formed. So if we wanted to be able to, if we have this kind of data, the goal for us is to take this data to be able to reverse engineer the underlying equations that describe this kind of system. This is exactly what we did previously for the ion velocity, right? We have the gene expression and the velocity with the known function f that map the gene expression to the velocity. So here, if we have the spatial temporal data, we want to be able to take this data and learn the underlying equation. But mathematically, this is much more difficult because in the original dynamo framework, we are basically learning an ordinary differential equation. It doesn't involve the spatial component. But here, because we do involve like the spatial gradient, the spatial diffusion for, in this case, BMP and the Wnt. So instead of learning ODE to model the spatial temporal kinetics, we will need the partial differential equations. So then I dig deep into the depths of literature and identify this method. This is actually very mathematically complicated approach, but here I'm not gonna go to all the technical details, but just show you the bigger picture ideas. So this method, the physics-informed Fourier neural operator approach, start with the measurement of gene expression. I mean, this is a general framework, but in this case, we can assume the input is gene expression V over the space X. And then you can first go through like Fourier, you can perform a Fourier transformation to generate different waves. And then we can take only the top modes. and then go through like involves the Fourier transformation, and then we can add this back to the original data after linear transformation, similar to the like the residual net, and then next go through the activation function. This is basically the key model behind this approach. And this method comparing to like other approach, which basically just solve an instance of Ph.D. This method actually try to map a function, map between function space. So basically, you can give like an initial state, you can map to the next state, and so essentially, this method is able to allow us to learn the Ph.D. function itself. So we here then generated a series of training data to try to demonstrate the benchmark this method. We started with many random initialization states, and then we follow this Ph.D. To generate as animation basically for this 3 gene over 10 and that's over space. We generate in total 1,000 simulations, and you can see on the panel D, right, if you plot the velocity and the Laplacian, basically the last term, you can see there is a very intriguing pattern for the BNP. And we can then take 80% of the data for training and the rest 20 for testing. So what I'm going to show you next are the result on the testing. So very interesting. You can see on the left corner, we achieve nearly perfect result. Right? The first row, that's the ground truth. The second row, that's the prediction. The last row is the residual. And for each heat map, the x-axis is the space, y-axis, that's the gene expression, and the color corresponding to the expression of a particular gene. So you can see from the second row in the first column, the spatiotemporal kinetics for the scRNA is nearly indistinguishable from the first row. I would want to emphasize that this is actually very challenging. This is a very difficult task. What we are asking for the neural net is that let me give you the gene expression state at the 10 zeros. So basically, give you this name at the very beginning, and ask the neural net to predict over 10 how this pattern will appear over 10. So in more intuitive terms, it means that if you know the weather at this moment for the Earth, can we predict the weather for the Earth for the next 1 week or 1 month? But as you see here, we are able to do a pretty good job. The figure shown on the right is a different method that was using the graph neural net. The purpose is, I didn't mention here, but the purpose is that in the single cell data, right, we are not talking about the continuous space. We actually each cell it's discrete. So that's why we use the graph neural net to represent this to model the spatiotemporal kinetics. So that's basically what I have. I want to end my talk with this analogy between like astronomy and biology for searching uniform theory. And I hope with the bigger data, more advanced machine learning, we are able to really gain predictive models for the biological system. So with that, I would like to give a big thanks to Jonathan and also my previous mentor collaborator, Jianfa, Yang Zhang, Cole, and many collaborators for their support over the years for me to work on all these very fascinating projects. Lastly, I want to take probably the last minute to share with you that since I'm starting my lab pretty soon, so if you happen to looking for postdoc other positions, please come to contact me. Yeah, we would love to work with you. Thanks.
All right, that was great. Thank you. Any questions for Xiaojie?
You know, I just had, I was really trying hard at the end to follow the physics-based Fourier operator, you know, the Fourier operator, you know, and part of it, I think, is when you said it was not a continuous function, but a discrete. So I could definitely see the digital Fourier transform. But I wonder what the physics was that was informing the Fourier transform. And I saw space and time. I get that part. And, you know, that sounds like physics to me.
The Fourier the Fourier laid itself, it doesn't involve the features constraint. So the constraint comes from the boundary condition. Like when you model this, right, you need to fix the boundary. Basically, in solving a PDE, right, you need to define the boundary, right? Like
that part I get.
Yeah. So here we basically that which is informed is defining the boundary condition in the operator.
All right, that's fair.
Yeah.
Yeah. Thank you. Sorry, I couldn't follow it.
Question. I was curious about the morphometric vector field you showed from the fly data.
Yeah.
When you were showing those alignments across time points, are you just like registering the spatial coordinates, or did you use the gene expression data as well? I see.
Because we need to assume cells similar. Yeah, the cells named should have similar gene expression, but at the same time, nearby cells should move to nearby cells at the next time point. So basically, you can use like a few step versus distance-based approach to jointly learn the minimize the distance between the gene expression and also distance between the distance for the neighbor cells. Yeah.
I see. Yeah, that's great. Any other questions? All right. Thanks again.
Thank you. Thanks a lot.